genomics, its use in health. That's exclusively what we do here, looking at your genetics or my genetics and predisposition to disease. Can you hear me at the back, sir? Are you okay? All right, and its use in health. What about using genetics or genomics in anthropology? Studying evolution and relationships between you and I, or between myself and other organisms. And there's a little bit of a test there of how much do you think your genome, how much of your genome do you share with a banana? Might sound silly, but think about it. Alternative views of genomes, the genome as an archive. And I think that's what we may be sharing with Fritha and Nick. You know, how would you see the genome as an archive? And hopefully by the end, what you may well be questioning is, whose genome is it anyway? I mean, the material that's sitting in you, the fact that it's come from elsewhere, it's settled in you, whose genome is that anyway? What about the concept of sovereignty of the genome? This is used in the world of genetics. And there's a phenomenal field of ethics around the formal study of genetics in health sciences. And then just think about this. I'm going to ask this question again at the end, is we constantly talk about evolution, and evolution is often how the environment impacts on ourselves or any other organisms and forcing it to evolve in a particular direction. The fact that we've now, for those of you who were at Mark Soames' lecture last week, you would have heard him talk about the brain and the development of the prefrontal cortex and how that makes us sentient and intelligent, and that part of our existence may in fact be very powerful in influencing how we evolve into the future. Okay, so those are just some of the concepts. So I'll talk to you about where the field of genomics has come from. Essentially, the field of genomics is a study. If this is a cell, sorry, how many of you are biologists in the audience? Biologists, I mean medical doctors or any level of biology. How many of you haven't even done matric biology? And that's important to know so that okay, very good. Well, there's my mark. I was wondering why we struggle with you, Steve. Okay, so if you have this concept, you know what the cell is, right? A cell in terms of a constituent of the human body. When we talk about the genetic material, we're talking about the vault of inherited material that is present in your nucleus, which just for the purpose of ideas, is at the heart of the cell. Okay, and within this vault, you have all of, let's say, 99.9% of your genetic material. Okay, and that would be the genetic material you've inherited from mom and from dad. And I'm saying 99.9% because outside of that vault, precious, whatever you have is sealed in there. Outside of that vault, in this yellow space outside of that vault, you would have what I call mitochondrial DNA. And these are tiny slivers of DNA that code for the energy generating machinery of the cell. Although they're tiny proportions of your genomic material, they're hugely important in generating the energy to sustain you as a human being. All right? So here's your genetic material. Your genetic material is made up by chromosomes, right? And these are your chromosomes. All of you, if you're human anyway, you should have 46 of these structures, which are your chromosomes. Okay? So 46 of your chromosomes make up your nuclear DNA. What's separate from your nuclear DNA? Your mitochondrial DNA, okay? So just remember that. So your nuclear DNA, and these chromosomes are made up of your DNA. So the DNA is this double helical structure here. The scientific name is deoxyribonucleic acid, and please don't take that name away. Okay? Deoxyribonucleic acid makes up your genetic material, which is housed in your chromosomes. Okay, so those are the structures. And importantly, the genetic material is your blueprint in terms of the plan of who and what you are as an organism. And this DNA, the business end of the DNA is the genes. Okay? So your genes are contained in this DNA, which are assembled onto the scaffolded structures called your chromosomes. And that's all mostly in your nucleus. Now, I just wanted to give you a concept of what a gene is and how they're arranged on the genome. So so as I said, you have how many chromosomes? 46, right? 23 pairs. If we presume this is chromosome 1, okay, just linearly illustrated, I simply wanted to illustrate that the genes are not equidistant apart. They seem like they're randomly strewn on the chromosomes. They're not random, because whatever gene I have, in whatever sequence along the chromosome, it'll be the same in you also, and the same in everybody. Okay? So if this was a chromosome called Africa, you're going to have a gene at the bottom of Africa called Cape Town. That's a fairly large gene. As you move up along the N1, you're going to have a very small gene here called Worcester. Okay, as you go further up there, there'll be a big gene called Johannesburg. And all the way up there in the north, maybe a gene called Cairo. All right, so that's the concept. There you get very different sized genes, because these genes are coding for different sized proteins. And that's really the business end of biology. As much as I will rave about DNA, the actual things that do business, the entities that are busy interacting with the environment, are mostly proteins. Okay? So mostly protein, either the structural proteins, whether your skin and collagen, or your bones, or your hair. I mean, those are the transactional units with the environment. So here are your proteins. They're coded for by genes, which are contained in your DNA, which is scaffolded on these chromosomes. So that's a simple concept. Any questions? Please don't stop. Don't hesitate to stop and ask me. So from this point of existence, here's a sperm fertilizing an egg. Okay, and what I want to simply illustrate to you, that from that point on, from the conceptus point on, if that is adequately embedded in mom's uterus, then what it requires is being fed with enough environmental agents. That's what I wanted represented here. Okay, sun, water, fresh air, nutrients from the environment. Ultimately, you would end up with a mature 9-month or 40-week-old fetus in mom, who will then give birth, and maybe 9 years later, you'll end up with this organism. Okay, it looks more or less like a human being. And with all of the tissues, remarkably, right? So imagine from that point on, where this just looks like a glob, you end up with something like this that looks vaguely human. I'm saying vaguely human because that's my son. So, but more importantly, this organism is made up with this highly specialized tissue, differentiated tissue, and you're getting nerve cells where nerve cells are meant to be, you're getting fat cells where they're meant to be, and importantly, your neuronal tissue where it's supposed to be. And that's quite an amazing thing. It's still, I marvel at this miracle. But the important thing for this talk is that from there to there, it's actually a very complex process. And this, if you can't see it clearly, it's purposely put up there for that purpose. Okay? It's a map of biological processes or biochemical pathways that actually illustrate for you how sugar is broken down, or entities from the environment are broken down, turned to energy and proteins, ultimately to produce this kind of entity. Okay? So this is a map of the biology that's going on within the cell. And although we know a lot about it, we certainly don't know everything about what happens. And from a geneticist or a biologist or a clinician's point of view, having mapped all of these pathways is hugely important, and we understand the complexity. And in this complexity, if anything can possibly go wrong, it usually does. And that's when we end up seeing people coming into our clinic, or whatever else, or psychiatrists will end up seeing them in their environment. Now, I've put this up there, and again, if you simply have this picture in your mind, these folk here in this concert are reading off sheets, right? And they're able to put together an amazing symphony, all right? Because they're being directed and they're running according to a program, and you're getting something like this kid I showed you earlier, something remarkable coming out on the other end. And it's beautiful music. But obviously, you need a reasonably skilled ear to be able to discern whether it's being played perfectly, or somebody's playing off key or off note, or whatever else you can call it. So that's the analogy I'd like to leave this picture with. So The Human Genome Project was conceived in about the early 90s, and this was a group of geneticists who got together and decided, sorry, am I moving around too much? And a group of geneticists who decided that it would be a fantastic thing to try and decipher the code that is embedded in our genomic material, okay? Because it is written, as you would have seen in about 3 or 4 slides ago when I showed you what DNA looks like. There's a code there, and the genetic code is made up of very simply 4 letters. And notwithstanding the complexity of what we all are like, ultimately there are 4 letters that constitute our genetic material, and that's A, C, T, G. I'm not going to tell you what they mean. It's not too important, and maybe that should be part of your homework. Just figure out what A, C, T, G means in genetics. And A, C, T, G are these base pairs. That would be an A there, C, T, G, or whatever else, and there would be a sequence of those being mixed up. And that code, as I showed you earlier on, depending on what that code is, a particular protein will be coded for by that code. All right? So the Human Genome Project aimed to determine what was the code starting from the tip of chromosome 1 all the way to the end of the X and Y chromosomes. Okay, you have 23 pairs of chromosomes, and 22 pairs are your autosomes, and again, don't remember this. These are your autosomes, but importantly, a pair constitutes your sex chromosomes, the X and the Y. If you have 2 X's, you're a female. If you have an X and a Y, the Y, importantly or not importantly, is what makes males male. Okay? And you'll come across this in a moment again. So the human geneticists want to figure out what the entire code was. Here, the A, C, T, G, what sequence were they, did they occur in, and where did the genes live next to one another. It was almost trying to figure out the telephone book of humanity. Okay, trying to figure out who lives where and what is their address and how you might contact that person. Okay, or where does that gene lie in the human genome. Very importantly, from the initial point of consideration of the human genome, a reasonable amount of funding was put aside for something called ELSI, or Ethical, Legal, and Social Issues Pertinent to Humans in the Human Genome Project. And again, you could read about this. And this is just a timeline illustrating from the early 90s what had happened with regards to the Human Genome Project, and it's completely deciphered now. But importantly for me is just illustrating that along with the Human Genome Project, a huge, well, part of the Human Genome Project was actually deciphering the genetic code of not just humans, but several model organisms. Now, why do you think they decided to sequence the mouse, Drosophila, which is a fruit fly, or a nematode, and a number of other organisms? Why would they bother? They called it the Human Genome Project. Any idea?
With the similarity.
Yeah. Yeah. So one, because they figured there's something similar about our biology, regardless of whether we're human or a mouse or whatever else, and I hope that will come out in a moment. But importantly, you could experiment to a greater extent with these model organisms. So you could breed mice in the laboratory to follow a certain trend. And you could dissect that mouse. Whether it's right or wrong, I don't know. But you could dissect it or, you know, and observe it doing whatever it's supposed to be doing. Right. So this is a really nice picture.
You have to watch the time.
Yeah, okay. So here's your quiz. So you share about 30% of your genome with yeast, about 90% with mice, believe it or not. Fruit fly, you share 60% of your genome with a fruit fly. How about human to chimp? You've probably seen this. Anybody? Quickly? Some more than others, right? Okay, so getting close to the end, Ernest Haeckel, I mean, he was quite an intelligent human being, lived from 1834 onwards, and he talked about the tree of life. He was more or less a contemporary of Darwin's, and in fact, came up with this picture. This is one of his original drawings, and called it Pedigree of Man, but man was up here, and he showed his way of thinking as to how man was related to all kinds of other organisms on the planet. And this was quite remarkable actually in his day. And you'll have a chance to see this at some stage.
Wow. What year was that?
This would have been probably about 1870-ish. Interestingly, he was a remarkable, but he made some serious blunders with talking about human beings as the separate species within the human race. And that was one of his worst ideas. This slide is simply meant to illustrate that if we have to start with an original entity, organism, with DNA like that, all of the variation you see on the planet at the moment has arisen because of mutations. So you would get a base pair, so the base pairs I talked about, a C would pair with a G, and A would pair with a T. So during the process of replication, producing identical bits, you sometimes get an error occurring, and human beings have a remarkable mechanism in their cells to repair any errors which creep in during the replication process. But sometimes, because you're replicating 3.5 billion base pairs with every cell cycle, errors creep in and they're propagated. That's called a mutation, okay? The origin of a new variation. And it mostly doesn't matter because not all of your genetic material is coding for proteins. Often enough, they'll occur, for example, in the Karoo. I had that concept of going from here to Cairo. You'll come across cities or genes, but somewhere along the line you'll come across a Karoo. And if there's a mutation that occurs there, or a tree falling in the Karoo and nobody hearing it, it's not significant. People in the Karoo don't take offense. But in reality that does occur. But if you have a mutation occurring in a gene that codes for a protein, you're likely to have problems. Okay? So just bear that in mind, and how mutations arise. If and when these mutations arise, so wherever you should have an A replicating opposite a T, if you end up with a G, there's a means where we can actually measure that if that's embedded. So that's how we track diseases in human families. A mutation may arise somewhere, many generations back. We try to map what that mutation is and how it might cause disease, because that mutation is then propagated from generation to generation and has an implication for disease. In terms of genetic variation, we know that humanity originated here in Africa. There's absolutely no question about that. There's no question from all of the genetic studies we've done to date that the greatest amount of genomic variation is present in indigenous African populations, reflecting the age of the human beings from Africa compared to human beings in any other part of the world. So the Smarties diagram simply illustrates a diversity of genetic variation here, and those people who left Africa in fact only took a subset of those variations. And as you can see, by the time they got to the end of South America, you've got much less variation than there is elsewhere in the world. So close to the end is we've got an enormous amount of genetic variation. I tried to explain how genetic variations originate and how they're propagated, and taking Haeckel's diagram earlier and imposing it right now on all of the species on the planet. I mean, what you have in each of us is this genetic code that we're carrying on our 46 chromosomes that in fact reflect the long evolutionary journey, actually from time dot there all the way to the time we end up looking and functioning as human beings on this end. So think about it as a voluminous archive that we're carrying in every single one of our cells. Again, this is by way of illustrating, again, just taking your mitochondrial DNA. What we know is there's a map of Africa behind this here. There's Africa all the way up there. There's Asia and there's Europe out there. The amount of genetic variation can be tracked in your tiny mitochondrial DNA, and we know that human beings in Africa have the greatest amount of genetic variation in their mitochondrial DNA, and here's where it's originated, and as we can see, as it diversifies across the planet. So we've done some work not so long ago, and in fact we're able to show that Madiba's maternal DNA, it derives from the original San population here in the populations of southern Africa. And in fact his Y chromosome, which is Dad's lineage, or the ultimate Adam, is in fact from the Great Lakes region of Africa. And this was a precious opportunity. We intended sequencing his entire genome, but we didn't. We couldn't get to him before he passed away. But very interestingly, all of the technology we have, in fact, recently has gone on to sequence the entire genomes of a handful of San people. Okay, and this was published in Nature. Many of you are familiar with that journal. And one of the people that was sequenced as part of this project was Archbishop Desmond Tutu. So this was really interesting in terms of showing the extent of genetic variation. So I'm going to stop my talk there, just having thrown ideas at you as to what the Human Genome Project is, what do we use it for, and how one might see it as a store of evolutionary information starting from time down. And these are the questions I had thrown at you earlier or at the start of my lecture. All right, thank you.
So I'm coming from a very different perspective on all this, being a creative artist. We have the privilege of being able to dip into a whole lot of different disciplines at our whim and translate them and throw things around without really being an expert in any of those fields. And for some time I've been interested in sciences and in the medical sciences. And I had thought maybe I should talk more neutrally in this presentation, but I think I'm going to talk from my area of interest and show you some of the work that I've done recently and how it relates to the themes that we have here today. But I think it's nice that Raj started with that image of the tree, because that's where I'm going to start my talk as well. And as artists, we're interested in images, in visuals, and the power that those images hold, and how they have the capacity to transform public perception. And science has many of these visual analogies that they run on, because all these complex ideas are being held through the visual. Be it the double helix or whatever. Anyway. But one of the most potent images in speciation evolution is this image of the tree, and it's been around with us for thousands of years, really. But it's still the image that is held when we talk about evolution. How do we understand relationships between things? Can you hear me at the back? Am I talking right? And actually, when we finish the talk, I'd like to come back to some of the iconography around genomics as well, and some of the metaphors that are held there, around the book and the archive and scaffolding and what these things imply. I think it's a conversation that we can also raise. So the projects that I've been involved with recently have been in museums, natural history museums, and there are two of them that I'll show you towards the end. But what my concern has been is how speciation is depicted in those museums as being something that is hierarchical, something that is linear, that isn't complexified in relationships between species. And I found sort of different ways of addressing that through curation and working with their displays. But my starting point is the idea of the tree. And as we know, this is Darwin's 1859 version that was published of the tree. But this was questioned, well, it's been questioned many times, but in this public forum in about 2001 by somebody called Doolittle, who wrote an article in Scientific American, in which it was called Uprooting the Tree of Life. And so this really brought it to sort of very public layperson debate, I suppose. About whether the tree of life was the most appropriate iconography to depict speciation and evolution, and in the light of discussions around microorganisms and the relationships between them. So he presented that actually the web was a more appropriate analogy to be using, because of firstly the length of time that microorganisms have spent on Earth, sort of more than 2/3 of any other organisms, and that there isn't any sort of verifiable way of creating a tree-like lineage from microorganisms. So it seemed an inappropriate metaphor to be using something that starts as a base point implying a common ancestor. That number one. And secondly, which many people have written about, but this idea of horizontal gene transfer or lateral gene transfer, that microorganisms transfer material sort of horizontally rather than changing from one species to the next. And I wonder if this is the next slide. Yeah. So this is the very common image that is now shown that illustrates horizontal gene transfer, so moving across rather than from one point dividing, bifurcating to the next. So it becomes more web-like in its form. So my interests were how this has not been taken up by natural history museums and in many other fora, that we still have this entrenched idea of the tree, like form in our mind, which if you take it to, you might say, well, why is that a problem? The tree, what's the problem with the tree? What is the problem with hierarchies? But I think what it comes down to is as soon as you accept the idea of hierarchies, you accept sort of divisiveness in species, in human species, in different population groups, and it extrapolated from there. And I think maybe that's where this has some import for the medical humanities. So this is Darwin's very famous image from his notebooks, where he was trying to find a way of depicting relationships, evolutionary relationships, and this is his I think diagram, where he comes up with this branching tree, and this is what most people accept was his logical conclusion. But he was working with a number of other possibilities as well, other visuals. And one of his famous examples, well, now becoming more famous, is his coral, because he worked a lot with corals and atolls. But this was one of the other possibilities that he had. And in fact, if you're reading his notebooks, he was more convinced by this idea of coral as a metaphor, as a visual metaphor for evolution, because the coral dies off over time, but it's still evident there. The dead coral still exists. It doesn't disappear, and yet it's dead, and it sort of grows through from one form to the other. Also, I think it has a wider base level, many of the corals, rather than starting from a single point. And it's much older. So it's sort of 790,000 years old, some coral communities, as opposed to the tree, which the oldest trees I think are 4,600 years old. So as a metaphor, it is actually infinitely stronger. And he said, I think the best image for this is coral. But then he abandons that and he goes back to the tree. So, and I think, and the reason for this is that the tree was a much more saleable idea, with an idea that was quite hard to sell perhaps anyway. I think he was relying on something, an image that people were very familiar with. And yes, of course, the idea of the tree goes through many cultures, but it's a very strong Christian icon as well. So going back, the tree being the body of Christ, bilaterally divided into two arms. Here on the left is an image from the 1100s. It's actually an image which is depicting everything that is known about the world, but it's through Christ's body, everything is laid out in this tree-like form. And then on the right-hand side, just another example of sort of genealogical tree of ownership, actually, land ownership. But these were the images that he was working with as references. And of course, there were other trees, like the tree of Jesse, which found themselves manifest in medieval cathedrals, multiple examples of trees. And here we have Haeckel's tree.
Wow.
So
it's exactly the same one.
Yeah, it is the same. It's 1866 as well. So I know. So the tree, it goes back a long way, and the tree together with the river are running through the Bible. They are the images that start off the Bible: the tree of life, the tree of knowledge in the Garden of Eden. As you end in Revelation, you have the river running, and there's a tree that appears as well. So it sort of frames the beginning and the end in biblical history too. So I mean, despite evolution Darwin's ideas seeming to run contrary to Christian ideas, I think he was very conflicted, as we know himself, and I think this is why this image was latched onto. So Haeckel's tree, as you say, it is interesting that he produced it at the time that he did, but at the same time it's a very deeply hierarchical image, starting with Monera at the bottom and moving its way up to man. Okay, we don't worry about the gendered aspects of that, but man at the pinnacle of the tree. That's absolute supremacy. And the form hasn't changed much when you look at sort of phylogenetic diagrams on the right-hand side. They start at a base point and they move up, they're ascendant, I think this is the main point of it. Moving upwards towards the heavens. They're aspiring. And the history of this is borrowed from Aristotle's ideas of the ladder, the scala natura, as well as the chain of being, sort of divine chains of being. And these they become taken up in medieval times as well and collapsed. So the image on the right, although it's from 1579, shows sort of very early ideas of divinity. It's moving from hell below, through the earth, through trees, through different simple animals, all the way up through layers of angels to God right at the top. So it's an absolute pyramid of divinity, which Haeckel's tree echoes almost precisely, putting man at the top of that tree. And then there's this 1745 diagram on the left-hand side, Charles Bonnet's idea, very, very similar, starting with air and moving up to man at the top in this ladder scaffolding idea, which, so that I'm interested then, coming back to the genome and the ladder of life and the scaffolding of life, how those words and images pick up on some of the ideas embedded within this. So the natural then conclusion from moving from that is into this progressive iconography. This is an image from the 1960s, very famous image on the top from no, it's okay. No, it's okay. So moving from crouching ape all the way to ascendant, forward-facing white European man. And this is one of Haeckel's images below. Yeah, that's what you were referring to. So the same ideas are embedded in that all the way through. And so that is the problem with the tree. So the tree too, I think, has been taken up because it's something people are secure with. If you think of it as an image, it's symmetry largely. It's something that's quite balanced. It's ordered. It provides shade and foliage. It's comforting. Whereas the idea of the web, juxtaposed against this, is chaotic. It's entangled. It's something that entraps. It's something that cannot be ordered. So it's a much more terrifying image for people to work with as an analogy. So although the idea has been around for a long time, in fact, in the 1800s, 1700s, a lot of early naturalists were working with web-like diagrams and not tree-like diagrams, but they were all abandoned at the end in favor of something that was more symmetrical. So in the museum too, and this is my interest, the hierarchies are absolutely picked up within the architecture. So the British Natural History Museum in London on the left-hand side, not only are the sort of hierarchies embraced, but it is a church. It becomes a cathedral of nature, organized in the same way as a church, with a central aisle with some naves on the either side, walking forwards up towards the light, towards the sort of ascending towards the sort of God figure at the top, I suppose. In this case, we have Darwin, actually, the little statue right at the top over there. And then on the right-hand side is the Museum of Comparative Anatomy in Paris, where you have the same sort of idea of walking forwards towards progressing into the future, the sort of march of progress, what Stephen Jay Gould talks about, the iconography of expectation. That civilization moves forward, and how this has sort of distorted the idea of where we understand ourselves. So of course it's also a Noah's Ark on the right-hand side, and I mean that's also my interest, is how these Christian metaphors are deeply embedded within the museum, be it about ideas of wilderness or the Ark or Eden on many levels. So these are more the way the museum the way in which we understand it. Things are put in separate categories, boxes, in separate areas. They form parts of the tree, units. They never really cross over. They have their discrete spaces. This is an extreme version of this, is the American Museum of Natural History in New York, which the top level is sort of the cladistic museum, in which you're literally walking through the different sort of clades and nodes of through a sort of evolutionary tree from point to point. Becomes enacted. As opposed to a museum like this, which is also the Evolution Museum in Paris, it's a newer version, was done in 1996, where animal specimens are totally overlaid. You look through glass, through mirrors, through reflections from one set of images and objects into another. There isn't any sense of a discrete space. And then visually one starts to make connections between different species. And the hierarchies, they're not entirely broken down in this way. So this is what my interest was in the exhibition, and I think you've got it as part of your reading pack somewhere, Subtle Thresholds 2009-10 in the museum, which was dealing with pathologies and infectious disease and how it's been represented, and looking at ideas of difference and analogy within that. But also how disease isn't seen as something discrete and independent of itself, and how disease is always relational. That an organism should be sharing something with something else, and it's about a set of relationships. So the way in which the exhibition was viewed and understood was activated by having to walk from one work to another and make connections between things to make any sense of this exhibition at all. So in that way, it became part of the web in the way in which you understood the exhibition, rather than discreetly walking around from one point to the next. And this was the problem a lot of people had with the exhibition. They wanted to know, where do we start? It's an interesting question for me, because it never struck me, but it's the tree. Where does this tree begin? It begins at the doorway, and then you walk through, and information comes to you as it opens out, which this probably just confused in circles. So these are just examples from that, using objects and images from their collection.
Sorry, where was it situated?
In the South African Museum.
Oh, it was in the
It is in the South African Museum. It was, you know the museum?
Yeah.
It was in that, going in from the rock art exhibit into the big open space, which has now been blocked off and used for storage. It was in that space then. Yeah, so, and there were things like this ruler, which was incrementally broken up with hundreds and thousands of different species names, and then a concordance, a biblical concordance of disease ran in one direction, and a more typical history of disease ran in the other direction. So these are different worlds intersecting and measuring a space, for example. I shouldn't go on too much longer. These were light boxes made up from pharmaceutical paraphernalia, which made different viruses. And so in the same way that I showed you the museum in Paris, I set up a lot of reflective surfaces, so you were looking from one space into the next, seeing the museum through different spaces, and sort of anthropomorphizing animals as well. Like that. And then this exhibition is on, more or less, still, about 15 months after it opened at the South African Museum. And so with my interest in speciation, and how do you take something in the museum and move it outside of its discrete space, I thought I'd work with the rat. Let's come back to the mouse. Why did people use mice to decode their genomes? But because rats are so very similar to us in so many ways, I think, is why I wanted to work with this. But as a species, it's so closely related to humans through medical, through psychological beings, and they live with us as commensurate animals. So the idea was to work with the empty spaces in the museum and populate it with rats, and or ideas around rats, and make connections to other cabinets and other themes within the museum. So bounce the idea of one species off many different areas. And yeah, as I say, it is, if you're interested, it is more or less still on. Some of the video isn't working, and the museum is sort of slowly collapsing step by step. So, yeah, might not be there by the time you get there. So working with the existing study skins, which reside in these cabinets, and different levels of museum labeling, it might not make sense to go into this now. But really, this is trying to break down that hierarchical structure and create different layers through which you understand information and the idea of speciation. So, and those are my Adam and Eve rats at the bottom, right-hand side, based on Masaccio's painting from the 1400s. Yeah. Okay, so that was it.
Hi, hello. I've got a completely impossible task, right? Thank you, Steve. Thank you, Susan. This is called being set up, right? So after Raj's amazing, compelling science, you know, just really like an amazing story, and after Fritha's amazing iconography, you know, leading us through the sort of the deep history of an iconography that recurs, you know, in these different contexts. It's just amazing. So I've been sitting there trying to think about what I could possibly bring to you, you know, that would be at all interesting. And I think I've got three things. I've been madly scribbling, right? But I think I have three things. And the first is it's an idea. It's a point of departure. So let's see how this goes. And what interests me here is the conventional separation, the binary that exists between medicine or science on the one hand and the arts on the other hand, right? And this is a binary. This is a separation, a dualism with a deep history, in Western modernity. The literary theorist Terry Eagleton wrote a great book in the 90s called The Ideology of the Aesthetic. And what he traces is what he calls the autonomization of the arts, right? Long word, autonomization of the arts. What he means by that is the way in which the aesthetic, the whole realm of the aesthetic, became severed from social and political and economic processes, right? It became something that sat on its own as art, outside of all of those things, and outside of knowledge as well. So we have the knowledge disciplines on one hand, right, concerned with the analytical, the empirical, so on and so forth. And then you have art, concerned with creativity, intuitiveness, inwardness, etc., etc. And what Eagleton wants to, what he remarks on is the double-edged nature of that process, right, of autonomization. So on the one hand, art is granted a kind of a license, you know, and you began by saying you have the privilege of dipping in and of not being an expert and kind of wandering into territory, you know, and being playful in a way, which is wonderful. So art is given that license. But then on the other hand, and this is the other edge of the sword, it doesn't matter. It's only art, right? Do you see that? How it's undercut? So I suppose this is the thought. This is the first thing that I wanted to bring. It was the idea of, instead of sitting and being comfortable with that binarization between science and art, what about mixing it up a little bit? What about thinking about the artfulness of science, if you will? In other words, the constructedness, the contingency, the saturation with metaphor and imagination. That's on the one hand, right? And what about thinking about art as containing forms of thoughtfulness, logic, knowledge, being in itself a kind of an archive, right? That would be to mix it up. If you did that, you'd be creating something new outside of that binary, a kind of a third space. And it may be, I mean, this is the thought in relation to this course, somewhat provocatively entitled Medicine and the Arts, right? It may be that the construction of that third space becomes really important for all of you. And I'll tell you why, because in the hierarchy of knowledge that operates in the academy and in society at large, art is always supplementary to science, right? So if you stick with the binary, art is going to illustrate science. That's how it is. It's going to be there, you know, to kind of prettify, but the science is the hard stuff. That's where it happens. And the art is where, you know, that's the soft stuff. That's where you get a little bit mushy, right? And I think probably you want to complicate that a little bit, yeah? So that's the first thing. That's the idea, right? So the second thing that I thought I could bring to you is a field of knowledge. And I mean, you'll know this already, so apologies. But the field of knowledge is it's often called STS. It's Social Studies of Science and Technology. Who's heard of STS before? Social. There. Okay. Well, that's cool. Now you have, right? Social Studies of Science and Technology. It's a big established discipline in its own right. It's concerned with often deep ethnographic study of the way in which science works, right? And there's some great lines coming out of the literature, and I'll share some of these with you. There's a sub-branch of STS focused on laboratories. It's called Laboratory Studies. Who's heard of Bruno Latour? Yes, okay, good. Now, a bit more familiar. A founder of Laboratory Studies. There's another marvelous scholar called Karen Knorr-Cetina, and I want to share some of her work with you. And she's interested in the performativity of laboratory space. We walk in, we're confronted by, you know, the paraphernalia of the laboratory, which carries in itself an enormous authority and weight. The space, the apparatus, the white coats, okay? What happens when we put on the white coat? We become a different being. You know, here we are, we don the white coat, we gain in stature and authority, we speak with a certain measure. We become knowledge agents of a very particular kind. So a lot of laboratory studies focus on that performativity. Laboratory studies are also interested in laboratories as epistemic sites of a particular kind. And they use an epistemology which is constructionist rather than positivist, right? And if I can sketch the difference for you very, very briefly, positivist epistemologies imagine that there are objects in the world and the job of science is to find those objects. But the objects pre-exist the science. They're there, like hard things, like fossils, right? And the job of the scientist is simply to find it, uncover it, like me, an archaeologist. Oh, look, I found it. Okay, and you find these things and science progresses. Constructionist epistemologies, on the other hand, are interested in the way in which those epistemic objects are themselves constructed through the interaction between the scientist, the knowledge agent, and the thing in the world. In other words, it doesn't pre-exist necessarily in the form in which it becomes an epistemic object, right? So science is about turning, if you will, natural objects into epistemic objects, objects of knowledge. And the laboratory becomes one of the prime sites of doing that. The other prime sites are the field and the archive, right? Prime sites where natural objects are turned into epistemic objects or are brought into culture. So the other line on this is around the enculturation of natural objects, right? And again, it's a kind of a conversation. Nobody's saying, that there's no such thing as genes in atoms, right? There's a material resistance, a presence there that is there. But the fact that we call them genes and atoms and the imagery that we associate with them and the particular manner in which they enter knowledge is part of this quite complicated conversation, right? It's not a simple thing by any means. I mean, that's okay. So while I was sitting listening to Raj, I was captivated by your language, right? I mean, this beautiful, rich, metaphoric language. The genome as an archive, genes as books, base pairs as letters of the alphabet, the vault of inherited material. That's wonderful, right? Vault, it's where you keep a treasure, right? The heart of the cell, the blueprint of who and what you are, a chromosome called Africa, biochemical pathways as a symphony, playing off key, the telephone book of humanity, the tree of life, the molecular clock, and so on and so forth. Right, so I understand that these were metaphors that you were using to explain complex science to a non-specialist audience. But I guess here's the question. Beneath the metaphors, is there something definite and hard and non-metaphorical? Or beneath the metaphors, do you find further metaphors? And beneath those, do you find further metaphors? And so on down. That's the question, right? And depending on how you answer that, you're either a constructivist or a positivist. Okay? All right, that's the second thing. Laboratory studies. I won't read Karen Knorr-Cettina's work. I'll see if I can find the paper and I'll share it with you. Right? There are a couple of nice sort of pithy papers on it. The third thing that I wanted to bring, and this relates to some of my own work, and it relates to the paper that you read on Kuru. The third thing I wanted to bring is a history of ideas, a genealogy, and it's a history of ideas linked to what I would call a discourse on human origins. And I wanted to say three things about the discourse on human origins in Southern African studies, right? And the first thing is this. It has very, very deep roots within Southern African studies, really deep roots. They reach well back into the late 19th century and the early 20th century. There was a great focus of work, right, around questions of human origins in disciplines like archaeology and anthropology and human anatomy and so on. There's this deep history of interest in questions of human origins, and those questions begin to form as a discourse, and central place in that discourse in Southern Africa is occupied by people described as Bushmen or San in the literature. Right? They occupy great centrality, and they're objects of intense scrutiny way back in the day. Right? So that's the first thing. The second point about this discourse is its entanglement with racial science. And the paper that I gave you to read tracks the work of Raymond Dart at Tuerafirin in the Kalahari, and a subsequent set of events, a group of San, displayed in the Empire exhibition and then later exhibited here in the Rosebank Showgrounds on what is now Middle Campus, right? And I understand that that's a complex and a complexly contested history, but it's deeply part of the disciplines of archaeology and anthropology. So in my own discipline of archaeology, there are two positions around this. The one position says that, racial science was kind of mainstream business in the sciences well into the 30s and I guess into the 40s, post-Second World War, Holocaust, a kind of universal horror at what had happened in Nazi Germany, and racial science kind of gets booted out of the academy, right? And so the one position within archaeology is that's the end of that stream, right? It's the end of an inquiry which then gets discredited, thoroughly discredited. And we move on and we do something different. That's position one. There's another position which says that sometimes it's not so simple. Sometimes things recur, right, as part of a kind of a deep inheritance, and reappear sometimes in new forms or in contemporary disguises. But that we need to, as it were, take on board that history in order to understand the complexity of that recurrence, right? I mean, that's the counterposition. The one is turn your back, the other is sometimes it's more complicated than that. So that's the second point I wanted to make about this discourse on human origins. The third and final point is that the discourse on human origins has become a post-apartheid trope, right? Do you know what I mean by trope? Tell me quickly, a trope is? It's like a meta-metaphor. It's like a big metaphor that organizes other metaphor. So a colonial trope would be something like Africa, the dark continent, right? And you say that, and then immediately a whole set of images and ideas kind of rush to fill that space. That's a trope. So in I think a fascinating way, the discourse in human origins has become a post-apartheid trope. And there's a story there about origins and identity and Africanness linked to genomics research, which I think is really, really fascinating, you know, and which brings questions with it, right? Okay, I think I'll stop there.
Perfect.